Welcome to BioBrainiacs. Today we'll explore the fascinating world of DNA, often called the blueprint of life. We'll learn what DNA is, how it stores information, how scientists extract it from cells, and why it plays such an important role in modern science and medicine. By the end of this presentation, you'll understand the basic science behind DNA extraction and why it's one of the most important techniques in biology. We'll also connect these concepts to the hands-on lab we'll be performing. DNA is the molecule that contains the instructions for building and maintaining living organisms. Nearly every living thing on Earth relies on DNA to store and pass on genetic information. You can think of DNA as a giant instruction manual that tells cells how to function, and without DNA, organisms would not be able to grow, reproduce, or carry out the processes necessary for life. DNA acts as a biological instruction manual. The information stored within DNA tells cells how to function, grow, repair damage, and create proteins necessary for life. It also determines many of the traits we inherit from our parents, such as eye color and blood type. Every activity that takes place inside a cell can ultimately be traced back to information stored in DNA. Although DNA was first discovered in the 1800s, scientists did not fully understand its structure until 1953. Rosalind Franklin's X-ray images played a crucial role in helping Watson and Crick develop the double helix model. In understanding DNA's structure, it transformed biology and opened the door to modern genetics. Today, much of medicine and biotechnology relies on discoveries that began with this breakthrough. DNA is shaped like a twisted ladder called a double helix. This structure allows large amounts of information to be stored in a very compact form inside cells, and the two strands wind around each other and are held together by chemical bonds. This unique shape is one of the reasons DNA can be copied accurately when cells divide. DNA is built from smaller units called nucleotides. Millions of nucleotides connect together to form the long DNA molecules found inside cells. Each nucleotide contains a sugar, a phosphate group, and a nitrogen base, and the arrangement of these nucleotides determines the genetic information stored within DNA. The sequence of these four bases creates DNA's genetic code. Just as letters form words, these bases form the instructions that guide life. Even a small change in the order of bases can affect how a gene functions, and an enormous variety of possible sequences allows DNA to store a tremendous amount of information. DNA follows strict pairing rules. These pairings help maintain the structure of DNA and ensure accurate copying when cells divide. Adenine always pairs with thymine, while cytosine always pairs with guanine. These predictable pairings are essential for DNA replication and maintaining genetic accuracy. DNA is wrapped around proteins and packaged into chromosomes. Humans typically have 46 chromosomes organized into 23 pairs. Packaging DNA into chromosomes helps keep it organized and protected inside the nucleus, and if it's stretched out, the DNA in a single human cell would be several feet long. Genes are specific sections of DNA that contain instructions for making proteins. Different genes contribute to different characteristics and functions in the body. Scientists estimate that humans have around 20,000 protein-coding genes, and together, these genes help determine everything from physical traits to how cells behave. DNA serves as the blueprint, while proteins do much of the actual work inside cells. Proteins help build tissues, transport molecules, and speed up chemical reactions. The information stored in DNA is used to produce proteins through a process called gene expression. This relationship between DNA and proteins is essential for life. Almost every cell contains the same DNA. However, different cells turn different genes on or off, allowing them to perform specialized functions. For example, muscle cells and nerve cells contain the same DNA, but use different sets of genes. This selective gene activity allows the body to create many different cell types. DNA extraction is the process of separating DNA from the rest of the cell. It allows scientists to study DNA directly and perform further analysis. DNA extraction is often one of the first steps in genetic testing and biological research, and without extraction, scientists would not be able to examine DNA in detail. DNA can be extracted from many living organisms. In classroom labs, strawberries are often used because they contain large amounts of DNA, and scientists can also extract DNA from human cells, bacteria, plants, and animals. As long as cells are present, DNA can usually be isolated using the proper techniques. Scientists extract DNA for many reasons. It helps answer questions about health, heredity, identity, and biological processes, and DNA extraction is used in hospitals, research laboratories, and forensic investigations. It has become one of the most important tools in modern biology. Thank you.